Today we welcome Professor Dr. Chase Beisel, the head of the RNA Synthetic Biology Research Group at the Helmholtz Center for Infection Research in Würzburg. He is a chemical engineer and biotechnologist and works according to the principle of a French researcher Louis Pasteur. In line with Pasteur's approach, I work in problems that both grant fundamental knowledge but also tackle social issues, with a goal to understand the diversity of CRISPR-Cas systems in order to be able to use them against genetic diseases or multi-resistant pathogens. Due to the corona pandemic, a lot of things have changed, and so we are conducting this interview digitally. Professor Beisel, could you tell us how your lab work changed with the corona pandemic, and are you maybe even doing research on a new corona medicament yourself?
Yeah, certainly. So my lab was impacted like pretty much every lab around the world. The university and the center had reduced reduced the number of people who could be working at one time, required space or more space between people in order to prevent spread. What that meant is that our capacity of the lab was reduced down to about 20% for about 2 months. And only recently have we been able to increase our number of people in the group. Thankfully, there was another lab that was available that we could start using as well. But it was certainly impacted through that time. Thankfully, everyone stayed healthy through it. We did think seriously, too, about what we could be doing to address the pandemic. And in that regard, we're actually working on a new CRISPR-based diagnostic platform that should help in the detection of SARS-CoV-2 as well as other viruses and respiratory pathogens. So we definitely saw an opportunity to apply what we do in the group to this particularly unique and global problem.
Okay, that sounds really interesting. As you just said, you're doing a lot with CRISPR, and also when we read through your publications, we saw that a lot of your work is based on the development of CRISPR innovations. What fascinated you when you first started to do research about CRISPR, and what still motivates you 7 years later?
Yeah, so I got into CRISPR through my background in RNA synthetic biology. I was introduced to the field as a graduate student. And basically through my graduate work, I came to recognize that RNA holds tremendous potential, but most of the things that we design in silico or based on rational principles ends up failing miserably the moment you stick it into a cell. And so that motivated me to study naturally occurring RNAs with the idea that if we can understand why these RNAs are conserved through nature, what they're doing, how they coordinate with other regulators in the cell, we could use those insights to better engineer RNA. And so through my postdoc, I was figuring out what I wanted to do as part of my independent position. This was back at North Carolina State University. And at that time, there was this explosion of RNA regulators that were being discovered. And my thought was, I want to focus on at least one or a few of these as the center of my research program to understand what they're doing in nature and then figure out how to engineer them. And one of the ones on the list at the time was CRISPR. At the time, a kind of quirky RNA-based immune system in bacteria. And I thought, that looks really neat. It can target DNA that nothing else can. I want to work on that. So that's what got us started. Admittedly, based on my experience with other natural regulatory RNAs, I thought engineering CRISPR was going to be really hard. Turns out that was not the case, which allowed my group to advance very quickly, as well as many other groups around the world. But that's what got me into it. And it's been, of course, an exciting, but also incredibly competitive, sometimes overwhelming field to work in. But it is amazing how much you can do with this immune system as a technology. And so in those moments when I feel like it's too much or I'm getting interested in other things, CRISPR just keeps pulling me back in.
You just mentioned that you got interested in RNA because you realized that it has incredible engineering potential. Was there like a special realization where you thought, where you first recognized that, or?
Yeah, so it was really only in joining the graduate group that I ended up pursuing my PhD in. So Christina Smolke's group then at Caltech. And I was definitely looking at different synthetic biology groups in the chemical engineering department. What I think really attracted me about RNA initially was how on one hand it's relatively simple in terms of the types of interactions it can perform, but that also lends to things like rational design just through simple base pairing interactions, for instance, and the relative ease in predicting its overall structure, particularly compared to proteins, made it much easier to jump into. And so those were some of the initial things that I really enjoyed and liked. It was a matter, of course, of translating those capabilities into actual functionalities inside and outside of the cell.
Okay. A lot of your work is based on the development of CRISPR innovations, if I recall so. And in your review from 2019 about barriers to genome editing, you stated that some cells can escape CRISPR-based editing, but it is not yet well known how they do so. Did the research about this problem progress, or do you have your own theories about this phenomenon?
Yeah, I would say it's still a very active area. And we definitely know more than we did even in writing the review about what some of these barriers are and how to overcome them. I still think the fundamental challenge, though, is how you use CRISPR efficiently in bacteria for genome editing, particularly as you go from one bacterium to the next. If you limit yourself to E. coli, there's actually a lot of really great things you can do now. But the moment you move into a non-model bacterium, whether it's something in your gut or can be used in an industrial setting or found in water, then suddenly all of those things we assume or are used to in E. coli get thrown out the window. And it almost feels like you're starting from scratch. And so if anything, we want to figure out what those barriers are and the extent to which you can figure them out ahead of time and have a way to get around them right away so you can actually start doing genome editing in that microbe.
And how would you address this? Would you just try to do some more research among more model organisms that, where you will actually implement the system later, or would you are you more trying to find underlying principles that might inhibit this process across many different organisms?
Yeah, admittedly, we would love to find underlying principles, call it the silver bullet for genome editing in bacteria, where you just have some standard tried and true approach that's going to work no matter what bacterium you work in. Whether that'll ever come about, time will tell, but I think now we're still at a place where you just have to dive into a bacterium to try and get it up and running. And I think something we've been pushing for the field and was communicated in that review is that you need to be communicating not only the successes, but also the failures, such as low editing efficiencies and figuring out, okay, why was editing so bad? What was going on? Can actually be really instructive. And I think this has been part of the synthetic biology mindset that you have this cyclic design, test, build process. And part of that is learning from your failures. And I don't think the field is doing that quite as much right now.
Thank you. Your working group also takes interest in exploring the mechanisms of CRISPR type 1 systems. Are there promising properties underlying CRISPR-Cas type 1 systems that set them apart from CRISPR type 2 systems? And we were just wondering, why is the CRISPR type 1 system in comparison to the type 2 system so underexplored?
Yeah, so there are some unique capabilities of type 1 systems that differentiate them from type 2 or Cas9. One of the biggest ones is that type 1 systems rely on this extra protein subunit called Cas3. And that's recruited to target DNA to nick one of the strands and begins degrading it in one direction. And so an analogy others have used is that Cas9 is a pair of scissors, whereas Cas3 in type 1 systems is a chainsaw. And so if you really want a good way to just destroy DNA, type 1 systems are the way to do that. And so in bacteria, it's a really great way to we'll use CRISPR as an antimicrobial where you can just destroy target DNA versus introducing a defined SNP that can be more easily repaired. But there's been a lot of really nice papers coming out really in the past year showing you can use type 1 systems in eukaryotic cells. And when you include Cas3, you can actually degrade extremely large segments of DNA. And that's going to open up some new capabilities and applications that type 1 or type 2 systems, aren't really suited for. And then your last point, why don't people use these all the time?
Yeah.
It's because they require anywhere from 3 to 6 proteins. And if you have to choose between that and one protein like Cas9, people are going to use Cas9. So that there is an energy barrier there, but if you can get over it, you do unlock some unique capabilities.
And when you say that the Cas type 1 system, rather you use the analogy of a chainsaw, don't you have problems to actually let the cell survive this editing approach when you just rip the whole chromosome or a big part of it apart?
Oh, certainly. So in bacteria, it's normally not being used for editing per se. You're actually using it to kill the bacterium and make sure they don't survive it. So in that case, it's great. Eukaryotes is a different story. And honestly there, I think this is one of the most immediate questions, is why does Cas3 just not destroy that entire chromosome and possibly kill the cell? Instead, it's limited to deleting certain size chunks. The size of those chunks seem to vary, but something allows the cell to survive. And so I think this could be the next round of research, is figuring out what's kicking in and why is that chromosome just gone? Or maybe the chromosome is gone in a certain percentage of cells. People just have never really looked for things that are dying out.
Okay. In our project, as one subpart of our project, we want to establish CRISPR without a Cas system using DNA-RNA triple helix formation. So Cas is quite a big protein with a molecular size of 128 kilodalton. So we want to ask you, did you ever have problems dealing with Cas systems because there was just so much on the plasmids? And do you think CRISPR without a Cas could have interesting applications?
Yeah, so certainly trying to introduce this very large protein or its genetic sequence for Cas9 or for type 1 systems, it ends up being a lot more. That has sometimes introduced complications. For instance, we've ran into these weird cloning issues trying to introduce nucleases into our constructs. Sometimes they go in very readily. Sometimes it takes a lot of work. And depending on the application, that can be a huge issue or one that once you have your construct, you're fine. At the same time, there have been specific applications highlighted in the field where smaller nucleases is better. So for instance, delivering CRISPR using viral delivery particles especially in mammalian cells, there are very real cutoffs that exist there. And some of the nucleases are just too big to go into whatever the length restrictions are. But that's also led the field to try and find ever smaller nucleases. And so there have been a report of some of these, I think Cas14 and some of these Cas12 variants are now approaching very, very small sizes compared to the traditional Cas9. So it's all to say that they're getting smaller, but as they get smaller, that certainly limits the type of applications you can do. If you could just eliminate the nuclease altogether, that would certainly be great. The challenge, though, is that the nuclease does provide some very, very real mechanisms that would be hard to achieve in its absence. So, for instance, the fact that the nuclease is responsible for unzipping double-stranded DNA and allowing the guide RNA to base pair, and then being able to distinguish even individual point mutations. And if you're relying on standard RNA-DNA hybridization, it's hard to have that level of specificity. So it's all to say that the nucleases provide some very real, tangible benefits. But if there are ways to eliminate them altogether and still achieve certain applications, that would be quite desirable.
Okay. You just said something about cell delivery systems you already developed, and we had one question about one you recently published about the DNA nanoclues, where you used that for to get therapeutics, also casts in the body. And these are like yarn-like DNA nanoparticles synthesized by rolling circle amplification. Why does one have the idea of creating a rubber band ball of DNA to deliver therapeutics?
Yeah, so for that one, I have to fully credit my collaborator at the time, Zhen Gu, who is now a professor at UCLA. And he specializes in drug delivery vehicles and coming up with more bio-based solutions. And so it was his group that had the idea of using DNA as the packaging material in itself and relying on that to not only package the nuclease, but get it inside the cell and then release the nuclease at the appropriate time and location. So I was primarily there to help on the CRISPR side and providing the expertise, but the genius behind that is all his.
Do you know of the reasons behind why to use DNA in the first place as a packaging material? Like especially, it seems a little bit like cells in the body should be a little bit averse to taking out foreign DNA that is in the body. So how does it work? Why does it work so well?
Yeah, I mean, on one hand, I would say there's still a huge need for well-developed and sophisticated delivery vehicles for gene therapy and other applications. And so that's still a really intense and ever-developing field. And so just having other options, other opportunities is a good thing. I think what motivated this in part was that when you introduce a delivery vehicle, depending on its chemistry, could end up being toxic within the body, or even if it's not toxic, it may stick around for long periods of time. And so introducing something that would be considered natural, like DNA, could be a way to circumvent some of those issues. In this case, I agree that you would think our cells would be looking for things that it would flag as foreign biological agents, and DNA like this might induce that. But at the same time, if you introduce something that's just so different than anything it's ever encountered, that sometimes is enough to avoid any sort of reaction. And at least from the initial tests that were done as part of that publication, there was nothing immediate that was coming up, but obviously more extensive studies and more advanced models would be necessary.
Are there more challenges that researchers still have to overcome when designing new drugs and thinking of how to actually deliver them to the patients, other than what you already mentioned with thoughts about toxicity and so on?
Yeah, so this is an area, admittedly, I haven't worked in as closely, and my collaborator, Zen, would know a lot more about this.
Okay.
But at least within the CRISPR field, there's still a push to be able to do genome editing in a human versus the more achievable approach where you can actually remove cells from the body or have separate cells that you can edit in tissue culture, and then you introduce them into the body. And putting something into the body directly introduces a whole score of issues, not only related to delivery and toxicity, but one of the things that have popped up recently is that many Cas9 proteins, which come from pathogens, many people already harbor antibodies against Cas9. And so by introducing this thing you're supposed to use for genome editing could actually induce a relatively decent immune response. And so people are trying to tackle that problem and figure out ways to avoid that from happening. So it's all to say is as people begin moving more and more into the clinic and tackling specific diseases and indications, these were some of the issues that pop up that have to be addressed. But I feel like people are working on this and finding good workarounds and moving forward on it.
Okay. As the time processes in this interview, we want to get to our next part. Another big part of our project is biological circuit design. 2013, you published a paper about the role of feedback in synthetic biology that also reviewed applications and future prospects of the field, like the development of systems with many different inputs, complicated logic modules like layered positive feedback loops, or designs that facilitate the optimization of circuits facing non-optimal conditions. Do you think there has been significant progress in these areas during the last few years?
Oh, certainly. And it's been fun watching as people have moved from developing these, well, what I would call toy systems, where you're basically controlling GFP, You're not going to cure cancer or anything else using that sort of approach, but it's a really great start to understand the basic principles. And so since that time, people have been able to build increasingly sophisticated circuits, bringing in new components like CRISPR. But on top of that, they've actually been working towards specific applications. And so some of the fun examples I've enjoyed, for instance, are using these sort of logic circuits for immunotherapy and CAR T cells, and using this as a way of programming when the cells are activated and when they proliferate to make sure that it's specifically recognizing cancer and versus anything else in the body. So that's merely one of many examples. I think more recently, and something I wasn't even thinking about at the time in relation to RNA, is that the use of RNA specifically in the realm of diagnostics is really reaching kind of a fever pitch. And not only is RNA being used for in vitro detection of different transcripts, nucleic acids, but they can start building in these sort of logic gates that people were playing with more for fun, and now they have very real application. So it's all to say that I think people are really moving along and developing well, and now these real applications where you can apply these sort of circuits are coming into their own.
And so how exactly would that look like when you say that combining RNA as a diagnostic molecule with biological circuit design?
Yeah, so for instance, if you're trying to design a diagnostic that's looking for a molecule or a set of molecules and wants to provide some sort of output, it's moving from saying we just want to look for this molecule and we want some output to say it's there or not, to being able to sense the specific concentration and producing gradients of color or other outputs, so you can look at it and actually back out the specific concentration, as well as figure out whether it's in a healthy or not healthy range. Some great work coming out in that regard, for instance, related to zinc sensing, specifically in blood samples from Mark Szynski's group. But on top of that, you can imagine also trying to bring in multiple inputs of not looking whether this molecule is there or this molecule is there, but for instance, asking, are they both there, or what ratio are they in? And then using that to provide a simple readout. And so that's where I think these circuits are really moving towards, being able to take in a lot more information and provide a real usable output as a result.
In a short comment you released in 2017, you stated that there is a threat of over-analogizing biological circuits with electronic circuits, which is maybe exemplified by efforts of some research groups, which, as you said, the fun prototypes, so like creating cellular memory-wise systems or systems that are able to count. Do you still think that the research community in this field is too drawn to the attractive comparison between biological and electrical engineering? And what are major features of biological circuits that set them apart from electrical circuits?
Yeah, certainly. So early on, there was this analogy between electric circuits and biological circuits, and I think that worked really well to illustrate or provide an analogy for what's happening in biology and how we can engineer it. And certainly we have this really great foundation around electronic circuit design that might map onto biological systems, but working on different organisms and their genetics, there is a fundamental distinction there. And sometimes if you try and stick with that analogy and that we are trying to map electrical circuits as we know it onto biology, that's when things can break down, especially as you try and interface those circuits with the natural processes, as well as the ability of the cells to replicate, mutate, and evolve. And so still imagine some setup where we can have circuits that really replicate the types of signaling cascades and other networks that take place naturally. And that would be another major thrust that I don't think has happened quite as much on the synthetic biology side. Within this, though, I don't know where I was going with this too. So in terms of what's happened in the field, I think people have taken on a more practical mindset, especially as they're trying to apply these circuits to specific problems. And in those cases, they're able to find things work, but now they have to integrate certain biological features or concepts, and it can't be purely surrounded in electronic circuit design. So it's all to say that I think in my mind, the field is moving towards some sort of happy medium that marries the concepts of electronic circuit design with what actually happens in biology. At the end of the day, that seems to be the most effective approach.
What do you think are major challenges the biologic circuit design community has yet to overcome to create truly unsupervised cellular circuits and maybe implement cellular circuits in human patients?
Oh, there's a lot there to tackle as part of this. The one I normally get stuck on is how much of basic biology we're still trying to understand. On one hand, that is a bit of an overgeneralization, and there are some things we know a lot better than others. But even in thinking about what's happening at the molecular level within a cell and how the cell is responding to any changes or actions, and how we can truly interface with that to have robust circuit design as well as signal processing is something that I think we're still trying to achieve. And the thing about biology that still dumbfounds me is the fact that you could have this autonomous machine that's operating on its own under varying conditions, but is able to go perpetually. And the ability to go in and mess with that and actually introduce something as we envision it, there's still a lot about that autonomous machine that we're still trying to understand. So I think that's one part. And then obviously going from an E. coli cell to a human, and then also taking into account the variability from one person to the next, not only their genetics, but their health state and other features, like that's hard. And so I still feel like there's a lot of knowledge and understanding to be gained to get there. But this is one of the fun things about synthetic biology, of being able to jump in, trying to learn these things, implement things, recognizing your failures, and coming back with something that's even better and improved.
Maybe building on that, how do you see the relationship maybe between the knowledge which is gained from trying to design these biological circuits and maybe failing a lot? Also, you mentioned that already. Do you think what should be a better approach when you're saying that there are still so many things that we actually are not able to understand yet, how these autonomous machines function? Would you see that there still has to be a lot of fundamental research on the model organisms that come from nature? Or would you say that maybe also more knowledge could be gained from just trying to build the systems yourself and seeing, well, what might be the problems?
Yeah, I mean, admittedly, I think all of those would end up benefiting or helping us work towards the end goal. And when you talk about working on different organisms, I think there's increasingly a push to move beyond even these model organisms and start getting into the non-model organisms and some of the real diversity that nature holds. And that's already been an incredible source of parts and components for synthetic biology. And so continuing to delve into those organisms and work with them, I think, could really provide a lot of really new and exciting insights. But at the same time, to really engineer these sort of systems, people have to keep working on trying out new designs, circuits, but be ready to embrace the failures and really dig in to figure out what went wrong versus, say, shrugging and just moving on to the next design.
Okay. That sounds like good final words. So thank you so much for joining us in this meeting. I think it was definitely we definitely gained some new insights, and yeah.
Thank you.
My pleasure.